Good morning, everybody. So in the last class, I think you guys have looked at the tutorial class on CNNs and MLP. We also have seen some demos, right, of how these networks work. So CNNs, obviously, you might have noticed that it's have created a buzz, a lot of excitement in the ML machine learning area, as they've been applied to a wide variety of visual pattern recognition problems. And they're very good at what they do. So part of the experiment is that CNNs have roughly emulate the architecture of the visual system. Because if you look at, it's not just like an MLP where input is a vector. Because in case of CNN, the input is an image, and even the hidden layers are not like 1D layers, like in case of a simple MLP. They are two-dimensional feature maps. So kind of like the layers of neurons in the brain. Because as you know, cortex is consists of 6 layers of neurons. They're all 2D sheets. And even if you look at subcortical structures, very often the neurons are organized as sheets, right? As sheets of layers of neurons. So and also another feature about a CNN is that a given neuron always looks at only a window of neurons in the previous layer. That is especially in all the convolution layers. Only when you go to the fully connected layers, a given neuron is connected to all the neurons in the previous layer. But so there are some features of the CNN even architecturally which seem to copy the anatomical features of the visual system. So there's a lot of excitement. But again, there are some features which are quite artificial because the training algorithm which we use to train CNN, the backprop algorithm, is quite artificial. I mean, how are you going to backpropagate the error from the output layer all the way to the hidden layers? Mathematically, it's okay, but biologically, it's not tenable. And another thing is there's a lot of difference between if you look at detailed anatomy of the visual system and CNN, there's no comparison. The visual system is so complicated, which rich organization both at macroscopic and microscopic level, and and the CNN doesn't have any of those features. So before we get too excited about CNNs, I think we should at this point take a look at the visual system, the mammalian visual system, because even humans and primates right have a lot of common features. The visual systems have lot of common features. But people study primate, macaque visual system to understand human visual system indirectly. So let us look at some aspects of the visual system. So like I was saying, one thing that is nice about CNN, right, is that it seems to create features with an increasingly larger scale and break up the problem, right, and create representations of increasing large scales as you go from input layer to the output layer. So if you look at the neurons in the first hidden layer, right, the response of these neurons seem to be tuned to simple oriented features like edges or bars and things like that. So these are the weight patterns of the neurons in the first layer. Now, if you go to higher layer, slightly middle layer, right, you will find neurons respond optimally to certain parts of a face. So this is a CNN that is trained on face recognition. And then if you go higher level, maybe the penultimate layer, there's the last hidden layer, you will find neurons responding to entire faces. There again, you will find neurons responding to a whole class of faces. So, if you look at the images in the last layer, so these faces are like kind of generic faces. They look kind of slightly ghostly because these are not a single face. They are like averages of many faces, whole clusters of faces. Therefore, they kind of look slightly ghostly and somewhat fuzzy. You don't see the crisp detail that you will see in case of a picture of a single face. Right, so like that, the neurons seem to be responding to local features, right, in lower layers, and increasingly more and more global features as you climb towards later layers. And you will see a similar organization even in the visual system, which is what this talk is about, right? If this is a picture of a macaque brain, right, and so input from the eyes first goes to this area called V1, which is the primary visual cortex. So it's the first stopover of first cortical stopover of the visual information coming from the eyes, and so if you look at even lower layers, if So it's not shown here. If you look at retina, neurons simply respond to dots. Dots could be either a bright dot with a black background or a black dot with a white background. Okay, so but whereas when you go to primary visual cortex, neurons don't seem to respond to dots, but they respond to oriented lines, just like the neurons in the first hidden layer of the deep network or the CNN. Then further on, if you go to V2, which is the next layer, right, you will find neurons responding not to bars, but combinations of bars. So, for example, two bars can form an angle, like this is an angular feature, or three bars can form some kind of a star, right? And so they seem to respond to those kinds of simple combinations of bar-like patterns. So now if you go higher up to inferotemporal area, so I think the visual system is distributed over. I mean, a big part of the mammalian cortex is is dedicated to vision. So it is spread over three areas because in fact all sensory systems are distributed over the posterior brain, that is beyond the central circles. But if you look at vision alone, it completely occupies more or less the occipital lobe, and also penetrates into the parietal lobe partly and into the temporal lobe partly. So it's a pretty large system. And so if you go to the inferotemporal lobe, the inferior part of the temporal, I'll go into all this in detail a bit later. So there you have neurons which respond to complex objects. So in this schematic, you can see this picture where and like neurons are responding to bananas and faces and Eiffel Tower and these kinds of complex objects that you see in the real world. So there's a progressive assemblage of features, right? Very primitive features in the lower layers, and as you go to higher layers, they respond to more complex features. Now, a deep network also shows that kind of overall pattern. So, thing is, so we can see even with this preliminary analysis of available data, so there in some ways, the deep networks are very good models of visual system. In some ways, are quite bad and quite unrealistic. So let us go take a closer look at the visual system because it's important to know because this course is not a course on ML. This is a computational neuroscience course. But my point is even a simple model like a CNN or a deep network that can be manipulated and massaged a little bit to make it like a good and effective computational neuroscience model. So how do you do that, right? And for that, you need to first understand uh, something about the visual system because in ML community, people don't care about how brain works. So that becomes a serious bottleneck. Whereas in computational neuroscience, people know too much about how brain works, but you don't know how to abstract it, right? So people try to create this super complex, detailed models which are very cumbersome to work with. Okay, so we need to know about a little bit about visual system. So visual system, right? The journey begins in the retina in the eye, because when light falls on the retina, it gets transduced into electrical signals by certain photoreceptors inside the retina, and those signals then are propagated further on by the optic nerve, which carries visual signals from the eye to the brain. And the first stopover of these signals is a place in thalamus called the LGN or lateral geniculate nucleus. Because thalamus is like a big, junction box. So most sensory input that goes into the brain first has to go through this junction box, and from there on, it is routed to specific parts of the brain. Like from there, for example, and thalamus is a much bigger structure. Within this, there are different ports to different sensory systems. So, for example, visual information goes via a part called lateral geniculate nucleus inside the thalamus. Similarly, auditory information goes via medial geniculate nucleus, which is another nucleus inside the thalamus, and so on and so forth. So, okay, so the information from LGN goes to V1, which is the primary visual cortex. That is the first cortical stopover. And from there, it goes to V2, and then MT and so on. There is one pathway. Then you have another pathway where it goes to V2, V4, and finally to it goes to temporal lobe to this inferotemporal area. So this is the broad architecture of the visual system. So in the picture on top, you can see more areas. So these are the primary visual cortex areas. So this is V1 is the code for primary visual cortex. So information from retina. So imagine there is a retina here somewhere. So retina is here. Okay. So from there it goes to this LGN. LGN has again several layers, and from LGN it goes to V1. From V1 it goes to V2. V2 has again several subregions. They call it thick stripe and thin stripe and so on. From there it goes to V4, posterior inferotemporal, central inferotemporal, and anterior inferotemporal, and so on and so forth. So there are several inferotemporal areas, right? This is one pathway. Then on the other side, it goes to medial temporal, right, and MST, and then 7A, which is again a parietal area, right, and ventral inferotemporal, inferoparietal, VIP, and so on. Lateral inferoparietal and so on. So there is one branch which goes from the V1 towards parietal areas, and another branch which goes from V1 towards temporal areas. Okay, so this is called the ventral pathway because this ventral is down and dorsal is back, right, up or back. So this is the overall architecture of the visual system. But think these pictures don't have too much sanctity because depending upon the level of detail you want to go into, you can come up with more areas and give more names, because in biology we like to give names to everything. We think that by putting labels on everything we understand how they work, but we don't, unfortunately. So there are also versions of mammalian or primate visual system architecture, or which identified something like 120 areas within the visual system itself. So the point is that doesn't help any further. Right? Just by having higher resolution and giving more labels is not going to do any good. You need to understand underlying principles. Okay, so these are some of the visual areas which we saw in this block diagram above, right? And so let us start our journey. How does the visual system process the image? So this journey starts in the eye, as you know. So light coming from the world and it goes into the eye, and then here this is a cornea. Cornea is a transparent, has a curved surface. So therefore light gets refracted already in this curved surface. Then it goes to the lens, which is another refracting medium. So again, it gets bent and focused. So this cornea is a fixed curvature. So you cannot, do it. It's not an adaptive thing. But lens can be adapted by controlling these muscles right here. It can be made thicker and thinner. Okay, so it focuses the image onto the retina in the back. And that's how you see the image. And particularly the highest density of concentration of light falls on this central part of the retina called fovea, which is where you have very high density of photoreceptors. So let us go more into that. So this in the picture here you see that so you see the retina and the various cells inside that. So you see these long rod-like features, right? And these are the photoreceptors. So you see the photoreceptors are stuck inside the wall of the retina. And there are two kinds of photoreceptors, rods and cones. So rods are good for night vision, and cones are the ones which are good for color vision. Right? Let me explain that more later. So then the information, so the photoreceptors convert the light information into electrical signals, and that goes to a next layer of neurons. So these are called the bipolar cells. So right here. And from there it goes to the next layer, which are called the ganglion cells. So you can see that here, ganglion cells. And from there it goes out, right, to the to the brain. So point is, retina has a very strange organization. So thing is, the first layer which processes light is the photoreceptors, right? But then, logically speaking, you would expect photoreceptors to be on the outer surface, that is somewhere here, right? You would expect photoreceptors to be here, so that light first hits them, and the response goes to the next layer, and then finally the output goes out. But that's not how it is. The photoreceptor is actually the last layer anatomically, right? And then you have the bipolar, and then you have the ganglion. Okay, so that is because when the light hits the wall, right, and there is something called the pigment epithelium, kind of darkish layer. So whatever light is not absorbed by the photoreceptors, right? If this is absorbed by this darkish layer, which can absorb this light. Um, so therefore, they are closely set inside the pigment epithelium. So this picture shows a more detailed organization of the retinal cells. So these are the rods and cones. So just conveniently, the cones are indicated by color. I mean, they're not actually colorful, but just indicated by color. So these are the red cones and green cones and the blue cones. So they are color sensitive because their response is a function of the wavelength. So you have different cones responding to different wavelengths. So this is wavelength and this is the response of different cones. So the rods are the straight structures which are not color sensitive. They have basically more a flatter response property. Right, and here again you have in each eye you have 120 million rods and 7 million cones. And fovea is the central region where there is a high density of cones but it has no rods. And cones are color sensitive, rods are not color sensitive. So if you look at the response here, this is the fovea. So 0 degrees is the direction that corresponds to the straight on. When eyes are looking straight on, that is the 0 degrees. Uh, so light coming straight on into the eye, right through the through the aperture, right, falls at this point, the fovea. And around this, if you look at the blue curve here, this represents the distribution or density of the cones. So they are high in the fovea, in the foveal region, and they fall rapidly away from that. Rods have a different distribution. Well, rods actually are almost absent in the center, but as you go to these peripheries, again, the rods are high again. And here there is again a small patch where you have neither rods or cones, because what happens here is if the ganglion cells are the last layer, in terms of organization in the retina. And the outputs of the ganglion layer, these axons, go outside the eye and then form a bundle called the optic nerve. So, and this is the bundle which conveys optical signals to the brain. So you might just consider this. For this to happen, the fibers from everywhere, so you have retinal ganglion cells closer to the inner surface of the retina, inner surface of the eye, right, all over here. And the outputs of this must come out and then must get collected in one place and then go out of the eye. So for that to happen, there must be some place where the fibers penetrate the retina and go out. Therefore, at this point, you cannot have any photoreceptors because you are making a hole, you're punching a hole through the retina. And in fact, such a place exists, and at that point, since there are no photoreceptors, you cannot sense light there. And this point is called the blind spot of the retina, of the eye. And usually we all have it, each eye has one such blind spot. We don't notice it as a dark spot in the visual field because what is a blind spot in one eye is not a blind spot in the other eye. Okay, so therefore you can see what is happening in that part of the visual field through one eye or the other. So therefore, and brain fuses the two images and you don't notice that. But through some careful experimentation, you can exactly locate where the blind spot is, and you can see actually that kind of a fuzzy spot in the in the external visual field, which indicates the presence of the blind spot. Okay, so you see the retina, you have these three layers. You have the photoreceptors layers, so consisting of rods and cones, and then you have right. So this is the pigment epithelium where the scattered light gets absorbed. Right here, the pigment epithelium. Then you have bipolar cells, an intermediate layer of neurons, and you have ganglion cells. Okay, now, so let us consider this. You have two eyes, and so this is the visual field, the circular arc that you are seeing here is visual field. And so if you look at so various points in the visual field, light from a given point goes to the eye, it goes to this lens, the central aperture, right, and falls on some part of the retina. So right from the right side goes to the aperture and crosses over this central line and falls on the opposite side of the retina. So light from right side falls on the left side of the retina. Okay, same thing is true for both eyes. Similarly, light from the left side falls on the right half of the retina. So we don't talk about the right half and left half of the retina. We talk about uh, the temporal side and the nasal side, because nose is somewhere here. Okay, so we call this the nasal. So this is the nasal hemiretina. So nasal hemiretina, half retina. This is the temporal, one side of your temples, right, and temporal hemiretina. Now, so here the black arc here is called the binocular zone, because light coming from this entire zone, okay, goes through both eyes. Whereas light coming from this zone, like this is the right monocular, that's one eye zone. This light cannot go into the left eye because the nose comes in the way. Okay, so it gets blocked by the nose, so it doesn't come. So this can be seen only by this eye. Therefore, it is called the right monocular zone. Similarly, there is a left monocular zone which can be seen only by the left eye, but not to the other eye. But there's a huge part of the visual field which can be seen by both eyes, and therefore it is called the binocular zone. Now, when you are looking at a point, right, especially when you're concentrating, if you're looking at hazily towards infinity, then right, that's different. But when you are looking at an object at a finite distance, then you fixate on that point. What that means is you direct your eyes so the eye has an axis. You can see that axis is what connects the central point of the lens, central part of the cornea, and central part of the retina. So this axis, right, is the direction which eye is pointed. So, so when you want to get a lot of information from an object in a visual field, you point both eyes towards that, so that the image of that object falls on the fovea of the two eyes. So you get maximum information about that, and that point is called the fixation point, or the fixation point. So you see that part of the information from the right side goes to the left eye and some to the right eye, and some part from the left. And information from the left side goes partly to the left eye and partly to the right eye. Okay, so both sides of the visual field, right, send the information to both sides of both eyes. So you have a pretty complicated situation, because how do you combine all this information, right? So that I get a unified picture of, because everything has now two copies, right? How do I get a unified picture of what is out there in the visual world? So to achieve that, brain does something really phenomenal. The way the wiring is reorganized and rerouted, so that all the information about one object goes to one place. So right now, all the information about one object goes to two places, which is quite confusing. Right. So for example, if you look at for the left eye, it's a nasal hemiretina. It has information about the left world or left visual field. So information from there. I can't see very clearly here. There's this thing missing. Okay, no, let us see. These lines are more clear. So let us take the right eye. So if you look at the temporal hemiretina, that is the right half of the right eye. This has information about the left world, right? And so fibers from here go further on. This is part of the optic nerve. But point is, there is a point here. There's again a big junction area called optic chiasm. Optic chiasm, where there's a lot of rerouting of these fibers take. So two optic nerves meet, and they kind of mix and exchange their fibers. And so then they kind of, some people go straight on, some people take a kind of a transit and get their direction changed and go to somewhere else. So the temporal hemiretina of the right eye, these folks go to optic chiasm and then take a turn and go to the same side of the brain. Okay, whereas if you look at the nasal hemiretina guys, these guys are able to see the right visual field. So these fellows send their fibers, which go to optic chiasm and go further on to the opposite brain, to the left brain. So same thing happens to the left eye also. The nasal hemiretina is able to see the left half of the visual field. So these fibers go straight on. The line is not very clear in the picture, but in the left, that is the left or the temporal hemiretina of the left eye, can see the right visual field. So these fibers go up to the optic chiasm, but then take a turn and go to the same side, to the same plane. So now you see what is happening here. Um, this is left visual field, right? Information. It is also left visual field information. These fibers and these fibers are now rerouted and recombined in such a way that the right brain has all the information about. So this is the right brain. The information is it is getting is from left visual field. Now, left visual field is available. Information is available in two places, right? Here, sorry, left visual is here and here, and both these kinds of information, source of information, are nicely recombined through this kind of brilliant rerouting of fibers, and all that is taken to one place. Same thing happens to the other side. So similarly, in the left brain, you have rerouting of fibers in such a way that all the right visual field information, so right visual field information comes here. Okay, so that's in fact it is even so. This is actually some the major stopover in the middle, and a lot more interesting things take place there. So let us look at that in a minute. So before we go any further, we need to get introduced to the notion of a receptive field. That is, we have seen that there's a you can have a visual object in the visual field out there in the world, and information from there goes to different layers of the visual system. I'm drawing an abstract picture. I'm not saying which is LGN, which is even nothing. Just think of them as a bunch of layers in a deep network. So what happens is at every level, a neuron in a given layer gets input only from a small window of neurons in the previous layer. It doesn't get to see the entire, all the entire previous layer. So therefore, same thing happens all the way to the retina. Imagine the first layer that you are seeing here is the retina, and then next layer could be maybe LGN, right? So then what happens in the retina itself, a given photoreceptor, a small patch of photoreceptors in a neighborhood can see only a small patch of the outside world, right? The part of patch of the image. Now, if you see, so this neuron then can only see a small part of the visual world, which is called its receptive field. It is the field over which it receives information is its receptive field. So similarly, this neuron now in the third layer receives information from this whole part of the visual field. Now, so each of these fellows can receive information over this part of the visual field. So you can see that neurons at a higher level, in a higher layer, can see bigger and bigger parts of the input image. All right, and in fact, you see that that's what was happening in a deep network, because in a LGN, CNN trained on face recognition. All right, you see that the lower layers respond to only small parts of the image. They respond to oriented features, whereas when you go slightly higher, they can respond to nose and mouth and other parts of the face. As you go higher, even higher, neurons respond to the whole face. So that is happening because neurons in this hierarchy, right? Lower neurons in the hierarchy can only see a small part of the image, and higher neurons can get to see larger parts of the image. Okay, so that is the overall organization. But what exactly do these neurons respond to? Right? What kind of features in the image do they respond to? So there is a progressive assemblage of properties. If you go to the ganglion cells in the retina again, which is the last layer, right? These are the ganglion cells. So this is a kind of a schematic that shows what kind of light stimuli elicit responses in a ganglion cell. So in this schematic, so what they do here is, this is eye, let's say of an animal, let's say of a cat or a monkey or something. So you shine light through the eye, and then so this is the animal's head. Okay, not a good artist, but so then you poke a microelectrode through this tissue into the retina, right, and record from the ganglions. This is how the experimental setup looks like. So in this case, so the thing is, you have to first identify if you take a ganglion cell here, or let's say I take ganglion cell somewhere here. Now that will respond to light coming roughly in that direction. Okay, because this is how the light travels in straight lines. So therefore, if I poke this point with my electrode, then I need to make sure that I need to get the correct direction. So I have to point my light source in the correct direction so that light falls on the retinal cell, somewhere in the region of the retinal cell, of the ganglion cell, right? And that's when I can record from it. So people, play around with that direction a little bit and then see exactly at what direction, right, at what angle you elicit maximum response from that ganglion cell. So here again, you can also play with the size of the stimulus, the size of the light dot that you're flashing on in that part of the retina. So if you look at the schematic on the right side, in the first one I'm showing here, uh, so they're showing a very small light dot here, and it shows a reasonable response. Right, this is then you in this case they show the light dot, but somewhere in this part. So in fact, when you did that, so this is the background firing. Okay, so this bar, dark bar shows the time during which the stimulus is presented. So before that, there is a background firing. It's firing all the time. But when the light is shown, it is a slightly increased firing. Okay, when the light is shown in this region. But when the light is shown slightly on this peripheral region, which is shown in gray color, um, the firing actually slowed down a little bit. It was almost paused, right? And after that, again, it picks up. You can even see a little rebound there, and then it picks up. When the light is shown in the center, right, but it's a much bigger dot, right, than this case, you can see that suddenly there's a high firing rate, right, and then after that, once you remove the light, it comes back to normal. Okay, now in this case, you see that the light is shown, but completely filling the periphery. Yellow is a light, so in this case, it fills the entire periphery. If you do that, again, the firing completely pauses for a little while. Then it picks up, and again, there's a rebound once the light is stopped, and then comes back to normal. So in this case, light is filling the entire receptive field, right? And then there's no change in firing. So by these experiments, from these experiments, you can see that there are regions where when light is shown, it increases the firing of the ganglion cell. Then there are regions, right, in the same around the same area where if you shine the light, it decreases the firing of the ganglion cell. Okay, so in this particular kind of a ganglion cell, which is called on-center ganglion cell, right? If you shine light in the central part, it increases firing rate. If you shine light in the peripheral part, it decreases firing rate. Now, the more you do it, the greater is the effect. If you shine a lot of light, with a large area in the central part, it increases the firing rate and takes it to the maximum. If you shine a lot of light in the peripheral part, it decreases firing rate and maybe even completely stop the firing rate. If you shine light everywhere, the two effects cancel out and there's no change. So cells which respond when you shine the light in the central part, that is, this is the receptive field. This is the area over which, right, the cell responds. Outside this, there's no response because it has no access to those points in the visual field. So similarly, there are off-center ganglion cells where if you shine light in the center, it slows down the firing. If you shine light in the surround, it increases firing. So therefore, logically, if you shine light all over the center, it completely stops firing. It shines light all over the periphery, this annulus, right? It increases the firing maximally. If you shine light all over the place, both center and surround, then there's no change in firing rate. So these kinds of receptive field pattern is something that you see constantly, right, in the visual system. And the idea of receptive field can be applied to other sensory modalities also. We may not have time to go through all that. But in auditory system, in somatosensory system, and so on, you have this concept of receptive field. So there are two kinds of ganglion cells, on-center and off-center ganglion cells. Now, okay, so that's how the ganglion cells respond, right? And that's the kind of organization they have. But with this kind of organization, what do they do to the image? Because what we care ultimately is how is the brain processing the image? We don't care exactly how the cells respond. We want to know how is the image processed and how are we able to identify objects in the image and things like that. So for that, let us do a small kind of a toy experiment. So let us say I have image of a zebra. It's a very nice black and white image. Now I take a receptive field. Receptive field is basically a weight vector, right? I'm taking a simple McClellan-Pitts neuron and with a weight vector. And the weights correspond to the central part is high and positive. Weights correspond to the peripheral part is low or even negative. So okay, so I take some numbers like that, and it's symmetric all around because our on-center ops around receptive fields are symmetric. They have same response in all directions. It's just that there's a variation radially. So here also we do the same thing. Only thing is, so this sum value here, right, indicates the sum of all the weights. So here the sum is 2.5, 2.6, right? It's still positive. Now, when I take a layer of such cells, such mathematical neurons, and apply it to the original zebra, this is the kind of response I get. It still looks like a zebra, so no major change. But just wait a second. Now I amplify the negative part weights a little bit and suppress the positive part a little bit, so that my sum has now gone down a little bit from 2.6. It has come to 1.5. You'll see that the zebra picture now starts looking slightly fuzzy. But now see what happens. I've made the weight so that the pluses and minuses nicely cancel out. In fact, you see in this schematic, that's what is happening. The plus and minuses perfectly cancel out in this schematic. I'm not saying every ganglion cell has exact same property, but this is a kind of a trend that you see in ganglion cell responses. When you see that, when you have a center and surround which perfectly cancel off each other, you see that there's no change in the response, right? No change in response when you give uniform illumination to the entire receptive field. There's no change in response. So we have taken constructed one such receptive field where plus and minus can cancel out perfectly. When we did that, you see that the zebra almost disappeared, but it didn't fully disappear. What is happening here is the so original zebra had pure black and pure white kind of patches. In this zebra, the black and white disappeared. Both black and white now has become gray. But at the borders, at the edges between the black and white, you see this stripes of pure white and pure black. Everywhere is now gray, but at the borders, at the edges, you now have this black and white stripes. So you see that this kind of cell is able to extract the edges, right, of the image, which is a very good thing because you need. So even if you look at a computer vision algorithm that processes images, one of the first things that it does is right improve the contrast and extract the edges. So then you process the edges and find the outlines and find and identify the objects and things like that. So this is a very meaningful thing that the ganglion cell is doing. We can kind of relate to what the ganglion cell is doing by using the simple model. That is obviously biologically not very realistic, but even the simple model can give you some insights into what the ganglion cell could be doing. So now I go further and make the sum negative. Then the response is like a negative of the original image, which is basically nothing very special. Okay, so that's what is happening inside the retina. Now you send the outputs of the retina, the ganglion cell, to the next layer, which, like I said, is a part of the thalamus, and thalamus is a big junction box, and most sensory information goes through different. These are like port of entries, right? So sensory information goes to different ports of entry, and before it goes to their corresponding cortical destination. So for visual information, so this is optic nerve. The port of entry is the lateral geniculate nucleus in thalamus. And from there on, the outputs of the LGN go to the primary visual cortex or V1. So LGN is a very interesting structure. So let us look at that. And so there are obviously there are two thalami, the two thalamuses, right, or thalami, right, one on either side. And therefore each thalamus has an LGN. And the LGN has 6 sublayers. And you can, for want of a better metaphor, you can think of them as the feature maps of your CNN. Because in the CNN also, each hidden layer has a bunch of 2D layers, right? So you can think of the LGN also as having multiple 2D layers, which it does. And so another thing is the 6 layers receive information from both eyes in very specific ways, because we have seen that all the left visual field information goes to the right eye, and that has already happened. By the time you go to LGN, you've already gone beyond the optic chiasm. Optic chiasm is here. Already the rerouting of fibers has taken place, and segregation of left visual field information, right visual field information has already taken place by the time you go to LGN. So you see that so these fibers are coming from the right eye, and these fibers are coming from the left eye. And these fibers now branch out further and go to specific destinations. I mean, like, very obedient schoolboys, right, going from the assembly line to the respective classrooms. These fibers very religiously go to their precise targets in the LGN. And how does this rerouting occur? So there are two sub pathways among the fibers that come from the retina. There are, so because I didn't mention before, but the ganglion cells are of two kinds: the magnocellular and parvocellular. So there's some magno is large and parvo is small. So there's some ganglion cells which have large cell bodies and some which have smaller cell bodies. So the fibers coming out of them are also kind of labeled separately and segregated. So the fibers coming out of the magnocellular ganglion cells constitute the magnocellular pathway, and those coming out of the parvocellular ganglion cells constitute the parvocellular pathway. So fibers of the so if you look at all these fibers, within that you have magnocellular pathway and the parvocellular pathway. So among these fibers, the magnocellular pathways or fibers go to layers 1 and 2. So layer 1 is here, layer 2 is here. So if you look at this guy, right, and similarly, the parvocellular pathway goes to layers 3 to 6. Okay, so there's a very nice rerouting here. Now, similarly, there are again inputs coming from the same side. So this is a bit of jargon we need to remember. Contralateral and ipsilateral. Contra means opposite, ipsi means same. So lateral means side. Okay, so contralateral means opposite side input. That means, so the LGN is on one side. So this, in this picture, it is the right side. This is right brain. Okay, so information coming from the left eye would be contralateral input for this fellow. Similar information coming from the right eye would be ipsilateral input. So input coming from the contralateral side, that goes to layers 1, 4, 6. And information coming from the ipsilateral side goes to layers 2, 3, 5. So there's a very explicit rerouting of fibers, and each fiber knows exactly where to go. I just want you to appreciate the kind of the precision with which these fibers find their targets and attach to the targets. Because obviously, I mean, I don't know if any of you have had the experience of putting together a circuit board, right? And if you want to, I mean, if you have done any electrical engineering lab or electrical circuits lab, you're very often asked to connect some circuits on a breadboard, and you have to take a wire and connect it from point A to point B, and or if it's a PCB, a printed circuit board, you can even get to solder fibers. So even a circuit which has maybe a dozen wires, right? It's so hard to make sure the connections are right. You make so many mistakes, and circuit doesn't work, and so on and so forth. Just imagine you have a million fibers in each optic nerve, right? And then these fibers go so precisely to the targets. And again, left eye targets go to certain layers, right eye targets go to certain layers, magnocellular fibers go to certain layers, and so on and so forth. This is really exquisite engineering, right? Biological engineering happening in the LGN. The same thing is shown in this some more simplified block diagram. So how different parts of the visual field go to different layers in the LGN. So this is the visual field on the top here. This is the right visual field. This is green region, green half of the ellipse, right? And part of the green goes to the left eye, part of the green goes to the right eye. Now the green from the left eye goes to layers 5, 3, and 2 of the left LGN, right? And then the green from the right eye goes to the remaining layers. That is layers 6, 4, and 1. So if you look at all 6 layers, they already have all the information from the right visual field. Okay, 3 from here, right side, right eye, and 3 from the left. It's a really beautiful organization. So similarly, there's a mirroring of the same pattern on the right LGN. So how are we doing on time? I think, yeah, we can stop at some time here. So LGN cells also have circular center surround receptive fields, similar to ganglion cells. They have a receptive field size of 1 degree diameter. We talk in terms of diameter because ultimately what matters is the directions of the input light, right? Not really the sizes or distances. So, like ganglion cells, right? Both LGN cells also have circularly symmetric receptor fields. They have either on center off surround or off center on surround, right? Two kinds. Then magnocellulose layers contain a mixture of cells with on off and off on receptor fields, and the paracellular layers contain, that is 3 to 6, contain only one or the other type of receptor fields. Okay, so also the surrounds of the LGN receptor fields have much stronger influences than the ganglion cell surround. So if you think of the intensity of the effect of the surround, so this could be something like, so this is plus, this is minus. So this is something like the ganglion cell. The same thing in case of an LGN cell will be, so both the negative and positive are much stronger. So this is a LGN cell, LGN neuron. Okay, they are weighted more heavily, and consequently information about the contrast is more amplified. So output from LGN goes to the V1. I think we'll get to this point in the next class. So I think we should know about visual system because only then we'll be able to know to how much of these details we want to put in a artificial computation model, right? Because that depends on the problem that you're looking at. You can take a simple CNN and pass it off as a model of visual system. That might be okay for certain purposes, but depending upon the question, you need to figure out how much detail you want to put into the model. So for that, it's good to know a little bit about the visual system. Okay, any questions? So no questions. So see you tomorrow.